want to welcome everyone back, and I also hope that everyone had a nice time last night. I think this is a great venue and a beautiful day today. We're going to start out today with our fourth session, which is tumor biology and rare nets, and it's going to be moderated by our Chrissie Thirlwell and James Bibb. And without much further ado, I'm going to turn it over to them right now. So thank you.
Brilliant.
Guys.
Thank you. Good morning, everyone, and welcome to day 2. I'm Chrissie Thirlwell, and this is...
I'm James Bibb.
We've been practicing. I've been coming to this meeting for 13 years now. I was reflecting this morning, and I have to say the lineup and the quality of the science and the discussions that we have every time we get together improves every single year, and I think it's a fantastic achievement and reflection of what NetRF has achieved over the last sort of 6 or 7 years or so. We've got a brilliant session lined up this morning. It's quite an eclectic mix, but really fascinating topics to listen to and discuss. So with no further ado, I'd just like to invite Pratik Singh. Oh, I've just overtaken from you, James. I'm terribly sorry.
Our first speaker is Pratik Singh. They're going to present their work on the insights into intestinal nets from an in vitro model to human EEC differentiation, together with Ramesh Shivdasani.
Thank you, James and Chrissie. So good morning, everyone. I'm Pratik from Shivdasani Lab, and this is a collaborative project with Joe's House Lab at Weill Cornell Medicine. Today, I'm going to talk about distinct transient states in enteroendocrine differentiation from stem cells and how this can be used to gain insights into intestinal neuroendocrine tumors. So we know that small intestinal neuroendocrine tumors are enterochromaffin-like cells characterized by expression of chromogranin A, synaptophysin, and serotonin. But unlike native EC cells, they are dysregulated and proliferative in nature. So to give an analogy, we can think of SINETs like bad apples and should be compared with good apples, that is the native EC cells. However, due to the scarcity of EC cells, traditionally people have been comparing SINETs with the whole small intestinal epithelium, which is like comparing a bad apples with a mixed bag of fruits. Therefore, we need to understand the differentiation landscape of EEC and use this as a reference standard to understand the steps dysregulated in the SINETs. In this regards, our collaborators from Zhao Lab developed 2D human intestinal cell lines. In these lines, engineered with an NGN3 ER construct. In these lines, addition of tamoxifen activates neurogenin 3, which is a master regulator of enteroendocrine differentiation. Thus, transient 2-day activation of NGN3 over here triggers a dynamic EE differentiation program over 6 to 8 days, where starting from 96 hours, we see expression of chromogranin A, which is a pan-enteroendocrine marker, and by 192 hours, more than 95% of cells are chromogranin A positive and start to express various peptide hormones. So now, to understand what are the sequential changes in the transcriptomic profile, we did a bulk RNA-seq across 2 independent lines and obtained around 4,000 genes that are differentially expressed during the time course, where at day 0, we see genes enriched for stem cell markers such as FOXQ1, SOX9, and MYC, and they turn off soon after NGN3 activation. And through a dynamic cascade, by 192 hours, we see various hormone transcripts marked in red over here that correspond to specific enteroendocrine cell subtypes. However, through bulk RNA-seq, we cannot distinguish the cellular states of these distinct EEC subtypes and what are the factors that regulate them. In this regards, we looked much more at a granular level using single-cell approaches. We did single-cell RNA-seq at day 1, day 3, day 5, and day 6 to capture the overall differentiation trajectory and identified 4 distinct terminal EEC subtypes based on the hormone markers they are enriched for, as shown in the UMAP. In the dot plot, we can see that all these 4 EEC subtypes are enriched for distinct set of transcription factors, which probably regulates their differentiation into these distinct types. Pseudo-time analysis revealed that all these 4 distinct terminal cell types originate from a single node marked by a red arrow over here that arises from the preceding 2 branches on the UMAP. To identify the cellular states of these preceding clusters, we grouped them based on the nearest neighbor identity and identified distinct factors that are enriched in each of these different states, where the terminal cells, that is X, D, and EC, are enriched for the specific hormones as underlined, and the preceding state from which they arise from is a NeuroD1 positive state. which is arising from either an ASCL1 enriched branch or a HES6 enriched branch. Overall, we see that the proportion of ISCs dramatically drops post-NGN3 activation, and by day 6, there is more than 50% of hormone-producing cells that are present over here. Thus, we know that transcriptional states are determined by chromatin accessibility, and in this particular case, neurogenin 3 activates a set of enhancers that leads to expression of cellular state-defining transcription factors. And this triggers a dynamic wave of cascade, further culminating in EEC hormones. We identified these specific enhancers using techniques bulk RNA ATAC-seq and single-cell ATAC-seq. And thus, after identification of these enhancers, we found the motifs that correspond to these specific transcription factors that leads to each of these cellular states. In the interest of time, today I'll be going only through the highlights of single-cell ATAC data. We found distinct motifs associated with each cluster over here, as we can see in the heat map, where motifs enriched in the ISC cluster, as you can see also on the UMAP over here, which is Fos-Jun motifs, is de-enriched in the terminal cell types, while terminal cell types starting from NeuroD1 share the motifs largely across the board, as shown in example MNX1 on the UMAP, where you see MNX1 is a factor which is expressed only in the EC cells, but its motif is shared widely. This is a correlation plot where the y-axis represents the genes for enhancers enriched in each clusters, while the x-axis represents the transcripts that are enriched in each of those clusters. As we can see, there is a higher correlation on the top half of the plot, specifically for the terminal cells. This means that the terminal cells largely have a similar chromatin profile, but with a distinct transcriptome. Of particular note, we found that motif for ASCL1 precedes NeuroD1 specific motifs. And ASCL1 is specifically more enriched in the EC cells compared to the NeuroD1 motif, which is more enriched in the non-EC cells. Overall, we identified distinct hierarchical steps in the endocrine differentiation and defined the cellular states and the regulatory factors associated with them. Here, the terminal cells are arising from a NeuroD1 positive state, which comes from an ASCL1 or an HES6 enriched state. Interestingly, various groups have previously shown that NETs in small cell lung cancer or prostate cancer are generally of two molecular subtypes, that is either ASCL1 enriched or NeuroD1 enriched. Since in our system we are seeing an ASCL1 preceding state before NeuroD1, we compared 13 in-house sequenced SINETs and publicly available 44 SINETs from Alvarez et al. With the bulk RNA sequenced EEC profile, where we see the terminal genes are enriched in the SINETs over here, but specifically lack the markers associated with ASCL1 state over here. Also, at the ASCL1 locus, the SINETs, which are represented in blue over here, are inaccessible at the ATAC locus and also lacks the K27 acetyl marks. On the other hand, NeuroD1 locus for SINETs show remarkable similarity with the normal EEC profile. Thus, to understand specifically the role of ASCL1 in this particular system, we CRISPR'd out the ASCL1 factor over here. And we see that post-ASCL1 CRISPR, in the ASCL1 CRISPR cells, the ASCL1 state is specifically lost, which suggests that this particular state was really dependent on ASCL1 expression factor. And once it's absent, all the cells are pushed through the other branch, that is the HES6, into the terminal state differentiation. But still, we found all the different terminal EECs being formed at different proportions. Specifically, at day 3, we see a higher proportion of NeuroD1 specific cluster, which is not there in the scrambled cells, which suggests that these cells are differentiating precociously. We corroborated with these with RT-PCR data and also have validated with several ISC markers. Further, we saw that on this correlation plot, as you can see, in the scrambled, the EC cells are very far away and distinct from the other non-EC cells. But post-ASCL1 CRISPR, they do get mixed up and lose their identity. Here, the bar plot shows, specifically as you see in the ASCL1 CRISPR, the proportion of unihormonal cells being expressed by these distinct cells has been reduced compared to the scrambled. which means these cells are expressing more specific hormones which is associated with state. As you can see in the bottom plot, where in the ASCL1 CRISPR, the EC cells is now expressing a mixture of hormones, unlike the scrambled, which is largely relatively enriched for EC cells. So overall, to summarize, today we have identified distinct intermediate cellular states during enteroendocrine cell differentiation. SYNETs are closely related to the terminal EC cells, but lack specific factors that are enriched in normal EC cells. And the loss of ASCL1 factor bypasses the ASCL1 state during differentiation, and this affects the enteroendocrine cell specificity. However, loss of ASCL1 alone is not sufficient to mimic the SYNET characteristics over here. Finally, I would like to acknowledge all the people who have contributed to work. Specifically Ramesh, who gave me an opportunity to work in his lab specifically on this project, and Neuroendocrine Tumor Foundation for making this work possible. Thank you.
Brilliant. Thank you very much. And you kept absolutely perfectly to time. So do we have, I'm going to field questions from the room, and James is watching online. So any questions from the room? Eric.
Very nice talk. I have a couple questions. What is the role of ASCL1 in normal gut development in animals? Has that been studied?
So as far as I've known, in the normal gut system, ASCL1 is not expressed in this differentiation cascade over here. So it's I've not known of a ASCL1 knockout mice in the specifically showing papers for the phenotype, but at least in our system, we have seen that it is not expressed in the gut in the mouse. But we know that it is expressed in the human system, and this has been also corroborated recently from Hans Cleaver's lab, where Joe Boomer has published a paper where they have seen in the human EC cells, they also have ASCL1, unlike the mouse.
Another question is, you mentioned that ASCL1 was not expressed in SYNETs, because there's a wealth of data published by other groups suggesting that it's highly expressed in GI neuroendocrine tumors.
Yeah, so if we looked at the data carefully, in our sets, as we have analyzed, as I said, it was 44 publicly available data sets and 13 from ours. And the transcripts for ASCL1 are completely zero. There is no transcripts, except a few. There are few outliers over there. So I'm not sure if the ones which were specifically looked by the others are those specifically outliers which were enriched over there. But those outliers are very few. Out of those 57 what we have seen, there might be only 7 or 8 which contributes to those outliers, which have ASCL1.
I was wondering how the delta cells that you were seeing compared to the ones that are normally in the pancreas, because the lineage seems to be quite distinct, and then whether or not why you're not seeing beta cells, which are normally very closely lineage connected to delta cells.
Oh, so the question is the delta cells you say, which I'm seeing, why is it not similar to the pancreatic?
Is it? I don't know.
So I think there might be a slightly miscommunication on my part. So this is all whatever I talk about. It was D cells, which is generally a cell type in the small intestine, and these are the cells which produce somatostatin highly, and nothing to do with the pancreatic. So all the cells which I spoke about are the small intestinal enteroendocrine cells, and nothing to relate with the pancreatic ones.
Okay. So then your somatostatin cells in that small intestine versus somatostatin cells producing in the pancreas, they kind of end up doing the same function, but I was just wondering, so you haven't looked at the transcriptome to say how do you get to the same final hormone?
Yeah, I haven't compared them with the pancreatic cells anyways, yeah. But compared to the mouse, I have seen that they look relatively largely similar in most of the transcripts that they are expressing, like HX and others, which are known to be responsible for D cells, and they are enriched for those specific transcription factors.
The issues here really, is there some it's not the issues, it's just that there must be some transcriptional factors that could be restored to get these cells to re-enter senescence. Is that the idea? These are a cultural system, you're characterizing these cells. Clearly there's some shifts with these, but is that the ultimate goal, and how would we get to it?
So that's the reason we developed the system, to use it as I said, reference standard for SYNETs. And now at this particular stage, we are comparing for what are the factors that are altered, and using that information, can we mimic that in this culture system? So starting over here, as I showed, ASCL1 was one of the factors which was completely absent. But just knocking out ASCL1, it's not dependent on those. But in other systems, as people have shown, specifically small cell lung cancer, that ASCL1 is crucial for initial development of the tumor, and then MYC comes in and drives this tumor further in evolution by expressing NeuroD1 and other factors, and gets into the neuroendocrine carcinoma phase. But so over here, I'm trying to say, apart from ASCL1, maybe these cells also need MYC overexpression or something like that to mimic those particular fates.
What was the role of tamoxifen? Because as you know, there's the papers now coming out and looking at sexual dimorphism and changes in ER receptor status. So I was just intrigued by that.
Very well valid question, and this is what was initial our doubts about as well. What is the role of tamoxifen? So I presented one Venn diagram, if you remember, where I said it was two independent patient samples. One was treated with tamoxifen, one was treated with doxycycline, and then we have looked at least at the 4,000 transcription factors which I showed is common between the both. So it is irrespective of the drug-induced effects over here that we have looked at. And those are the ones which we are also seeing from the single cell analysis, which are correlated over here.
Brilliant, thank you. It's really, really impressive work, so thank you very much.
I appreciate it.
So next we have Qianjin Gao and Patricia Dahia going to be talking about human and murine single-cell RNA-seq in relation to Pheochromocytoma. So over to you.
Hello and good morning, everyone. My name is Qianjin Guo from UT Health and Patricia Dahia Lab, and the title is Human Murine Single-nucleus RNA-seq reveals potential cell lineage mechanism for TMEM127-mediated susceptibility to pheochromocytomas. So adrenal is on the top of kidney, and it has two parts. One is adrenal cortex, and another one is adrenal medulla, and composed of chromosome cells, and which can produce catecholamine like epinephrine and norepinephrine. And the pheochromocytomas is real tumor derived from adrenal medulla, and clinically the symptoms could be like high blood pressure and rapid heartbeat. Genetically, there are a lot of the genetic drivers for this tumor, and the TMEM127 is one of them. This gene was identified in our lab around 2010, and it is reported to have 4 transmembrane domains and acting like as a tumor suppressor gene, but the underlying mechanism is still not so clear. So our hypothesis here is cell-specific transcription profile of adrenal linking TMEM127 probably will provide insights into this mechanism, which could lead to the pheochromocytoma development. So yesterday we talked about a lot of the mouse models. So we have the same situation here. TMEM127 knockout mouse didn't show any pheochromocytoma phenotypes, but I think it's provided us an opportunity to look at what happened at the very early stage of the tumor genesis. So we used the frozen adrenal gland from wild type and knockout mouse, and also some frozen human cells to do the single nuclear analysis. So use a lot of the downstream RNA packages to infer the cell type ID and the proliferation potential, and also copy number variations, and a lot of the function analysis. So here is a typical UMAP of mouse single nuclear data, and it's split by the genotype. And we can see from the UMAP here, there are no specific cell cluster in each genotype. And based on different cell cluster markers, and we identified 19 cell clusters here. And of course, we are interested in the chromosome cells here, and they all express some common markers like TH, CG, and CGB. Well, according to the different expression of the PMT, we divided the 2 chromosome cells into 2 clusters. One is immature chromosome cells, another one is mature chromosome cells. So PMT is an enzyme which can convert the norepinephrine to the epinephrine, where cells is more mature. And so the mature chromosome cells usually shows higher PMT expression level and secreting the epinephrine, while the immature chromosome cells usually shows low PMT expressed and secreting norepinephrine. So we are wondering if TMEM affect equally on the 2 chromosome cell clusters. We look at the cell population and found that only the mature chromosome cells is highly abundant in the knockout mouse. Well, there's no difference for the immature chromosome cells. And when we look at the cell cycle phase, and very interestingly, only the mature chromosome cells show higher G2M and S phase, which means the knockout mature chromosome cells shows higher proliferative potential. the TMEM127 affects more on the mature chromosome cells, not on the immature chromosome cells. It's relevant to the human disease since the Pheochromocytomas roughly divided into two clusters. One is the cluster 1 is pseudohypoxia, and which is featured by secreting non-epinephrine and with a low PMT expressed. While the cluster 2 is a kinase signaling group, and which is featured by the secreting the epinephrine and with highly PMT expressed. While the TMEM127 mutant Pheochromocytomas were just belongs to the cluster 2. So we are wondering how to find the driver genes, driver genes behind this process. So we use the regulatory network analysis to find 5 specifically activated transcriptional factors in the mature chromosome cells, and among which only the EGR1 is highly expressed in the knockout mouse. And also, we confirmed the EGR1's location in the mouse adrenal is located in the adrenal medulla, not in the cortex. And also, we validated our finding in the human data, and we used the microarray and also the Western blot to confirm EGR1 is all highly expressed in the TMEM and other kinase-related tumors, which may indicate that EGR1 may play a common role in the kinase-related tumors. So next, we want to look at the human data, and then this we take 2 typical kinase-related tumors, and one is TMEM127, and another one is rat. And also, according to different cell class markers, we identified the 10 cell classes here, and for the chromosome cells, we got 4 subgroups. And very interestingly, for the cluster 1 and cluster 4, which were shared by the 2 mutant tumors. Well, for the cluster 2, which is more enriched in the TMEM127, and for the class 3, it's more enriched in the rat mutant tumors. So since they are all tumor samples, we are wondering if they are the true tumor samples. And we did the combinatorial variation to identify the gain or loss of the chromosomes. And interestingly, almost all the chromosome cells here shows the combinatorial variations. And also, we can see the chromosome 1p and chromosome 2q, the loss is reported like chromosome 1p is common to the rat and TMEM127 mutant, while the chromosome 2q is reported to here, consistent with the previous report, it's in the TMEM127 mutant tumors. So since we got the 4 subgroups of the chromosome cells, so we are wondering the difference among the 4 clusters. So we found very interestingly the cluster 4, even they had very few cell populations, but they had a lot of the cell cycle marker like the Ki67 and TOP2A, and also some predictor markers. And for the cluster 1 here, it had some markers that shows it's the early chromosome cells. Of course, they all expressed the common chromosome cell markers like TH and so on. And then we try to evaluate the differential state of each 4 subgroups. And interestingly, the cluster 4 and 1 shows the least differential state, while the cluster 3 from the rat mutant shows the most stable state. Then based on this, we did the stool time analysis, and we found the 2 tumors actually seems like start from the same cell orange and then developed into 2 different directions, one to the rat direction and one to the TMEM direction and one to the rat direction. So as we discussed about the DGR1 may probably play an important role in these kinase-related tumors. So we also look at the EGR1 features in this human data set. And interestingly, EGR1 is mainly located in the shared clusters, cluster 1 and cluster 4. And also the signature from the mature chromosome cell from the knockout mouse is also enriched in the shared clusters, the cluster 1 and cluster 4, which suggests that maybe the EGR1 play a conserved role from mouse to human, and also may play a role on the very early stage of the tumor genesis. So this is about the results, and for the conclusions here, we found the TMEM127 knockout adrenals had more mature chromosome cells, And the regulatory signature of knockout mouse, knockout chromosomal cells shared by the kinase-related tumors. Also, the human tumors of kinase cluster maybe probably share the same cell orange, and also EGR1 plays a role in this whole process. So next, finally, I want to thank all the lab members in our lab and also all the support from our collaborators, and also importantly, thanks for our fundings, especially for the Neuroendocrine Tumor Research Foundation. Lastly, thank you all. Happy to take your questions.
Are you planning to manipulate EGR1 in your system to validate whether it's playing an important role? What are your next steps?
Yeah, it's a good question. The question is, what's our plan on the EGR1, this gene? And actually, we probably may to check, use the CRISPR to knock out EGR1 to see the cells, like the proliferation or the other state, but it's maybe not done yet. It's maybe next plan for this. Yeah.
Justin?
Your population 4 was kind of defined by the cell cycle expression.
Also the cell proportion, like the percentage of to the total cell numbers, the two.
I see.
Yeah.
Do you think that that's a transient state, and then those cells are moving into another more stable state? Like, so those are, when they replicate, they're going there, and then they
Yeah. A good question. The question is whether the cell population difference is due to the transit from one cluster to another cluster. Well, actually, the total chromosome cell, the proportion is not changed. So we don't think, we have not evidence to say it's a transit from another cluster to another one. So maybe it's the effect specifically on the mature chromosome cells, not the transit from one to another. Yeah.
And then the other question is, it was a Cree-lock system. I didn't see what your driver was. Maybe I missed it.
Oh, it's a whole body knockout. Yeah.
In connection to that, these animals don't actually get pheochromocytomas, but they are vulnerable to things like renal carcinomas. Are those happening in these animals?
Actually, the phenotype, we already have publication, but on a mouse model, but it's not pheochromocytoma related. It's like insulin sensitivity and the weight loss. So no phenotype to the PPGL found yet.
Yesterday we had some very constructive discussions between the gap between clinical concerns and the challenges of basic research. And towards bridging that gap, our next talk on mesenteric fibrosis in small intestinal neuroendocrine tumors is going to be presented by Maria Martins and Harry Hodgetts at University College of London in the UK.
Hi everyone. So we would just first like to thank the Foundation for the opportunity to present our data here today. So I'll start with a bit of background on mesenteric fibrosis, or MF. So MF affects up to 50% of patients with SYNETs. It's a very common complication, and this occurs when there is a fibrotic reaction around the metastases in the mesentery, which can then lead to the development of a larger mass, as you can see on the CT image on the right. And this then leads to other severe complications in patients and also a shorter overall survival, as we see in this graph. One of the main problems is also the lack of understanding of the pathophysiology, which limits the development of new treatments. And so this is why we have this project funded by the NetRF, which is a collaboration between two eNET centers of excellence at Royal Free Hospital and Erasmus MC. And as you can see on this diagram that shows the overall plan for our grant, we are focused on understanding the pathways involved in the carcinogenesis and fibrogenesis of MF, also identifying biomarkers for the diagnosis and prediction of the clinical risk, and also evaluating the predictive role of radiomics in looking at the clinical outcome in these patients. So in orange, we have highlighted some of the steps that we have already completed, and in yellow, those that we will be discussing here today, focusing on the co-culture experiments and the analysis of patient tissue that has been carried out in both centers. Starting with UCL, we have analyzed our first cohort of patients, which is composed of 45 individuals, for which we have normal intestine, primary tumor, and mesenteric mass. And these patients have been classified into 4 different groups of fibrosis severity according to histological, radiological, and surgical assessments. So from these tissues, we have extracted RNA, we have performed RNA sequencing and analyzed it using gene set enrichment analysis, or GSEA. We have also used this RNA to look at the expression of 5 transcripts that we have termed the NET test fibrosome markers. And these have previously shown by our group to detect MF when analyzed in the blood of SINET patients. And these were originally derived from the NET test developed by Professor Modlin. So we have used RT-qPCR to look at their expression in the tissue. Starting with the RNA sequencing analysis, we have performed the GSEA for 25 comparisons that we have between the different fibrosis groups and the different types of tissue. We have then screened for significant reactomes related to fibrosis and extracellular matrix, and then created a list of target genes, including the core-enriched genes that were significantly up or down regulated within those reactomes. This is just an example of the data that we obtained from the GSEA. So we have a large amount of data, so we cannot show everything here today. But if we focus on this first comparison, which is between the mesenteric mass in minimal fibrosis group and the mesenteric mass in the severe group, we have a positive enrichment in the extracellular matrix reactomes and also in the carcinoma-associated fibroblast reactome. And in general, across the different comparisons, we have seen significant positive enrichment in fibrosis and ECM-related reactomes in the mesenteric mass compared to other tissue types, and also in the more severely fibrotic groups. In relation to the NET test fibrosome markers, we have seen an upregulation of CTGF and NIP3L in the mesenteric mass compared to both the normal tissue and the primary tumor across all fibrosis groups, and also an upregulation of CD59 in the mesenteric mass compared to the normal tissue again. However, for APLP2, we saw a downregulation in the mesenteric mass and also downregulation in the primary tumor compared to the normal tissues in all fibrosis groups, including the non-fibrotic. And we have seen no significant changes for FRISL7. For our next steps, we'll be taking the selected targets from the RNA sequencing and looking at them further in vitro cultures to understand their impacts on the crosstalk between the cancer cells and the fibroblasts. And we will also be developing a 3D culture model using gels made from the ECM of decellularized human intestine. And we'll also be analyzing the DNA methylation in the patient tissue of our first cohort that we have mentioned before. So now I'll hand it over to my colleague, Harry.
Thank you very much, Maria. So I'll be presenting a few slides of ongoing research from our collaborators in Erasmus, who unfortunately could not be here to present today. So they are currently developing 2 models to assess the interactions between SINET cells and cancer-associated fibroblasts. The first being a 2D monolayer model looking at paracrine signaling. So this involves taking GOT1 cells and taking conditioned media from them, and then using that to culture primary CAFs isolated from SINET patients. And as part of the optimization of this method, a BRDU was performed, and as you can see by the graph on the right-hand side, the proliferation of the primary CAFs increased significantly when they were cultured in the GOT1 conditioned medium, compared to when they were cultured in a control medium containing no serum and 0.1% BSA. The next steps of this model will be to isolate and culture primary SINET cells, and again take conditioned media from these, and culture primary CAFs from patients with mesenteric fibrosis or without mesenteric fibrosis, collect the supernatant, and then run proteomic analysis, firstly looking into the pathophysiology, but then hopefully developing that into potential biomarkers and drug targets for mesenteric fibrosis. The second model being developed at Erasmus is a spheroid 3D model. This is again using GOT1 cells and also using fluorescently labelled human pancreatic fibroblasts and growing them together as a co-culture in a single spheroid. These 2 cell types are already known to interact with each other and so are being used to optimise and validate this model. And after these spheroids were grown, they were dispersed into single cells and analysed using FACS analysis. And as you can see by the graph on the right-hand side, the number of HPF cells increased significantly when they were co-cultured with the GOT1 cells in a single spheroid compared to a HPF-only spheroid. So the next steps from this model is to again take the supernatant of the different types of spheroid and run proteomic analysis. and also to perform histology and immunohistochemistry on the different spheroids to better assess at the cellular level the interactions that are happening between the cells when they're in close proximity to each other, with the ultimate goal of isolating and culturing primary SINET spheroids and co-culturing those with the CAFs isolated from SINET patients. So the next steps of this program are highlighted in green, and a few of these we have talked about already, including the secretome analysis, the target gene analysis, and the ongoing epigenetic study. We will also be performing a retrospective follow-up study based on previous data published by Erasmus on the role of sex steroids in the prevalence of mesenteric fibrosis, and also a follow-up study using combined cohorts from Erasmus and the Royal Free Hospital on the predictive role of radiomics on clinical outcome in mesenteric fibrosis. And we'd just like to highlight at this point that we're focused on working really well as a collaboration together to enhance the data we receive from each group, and that's achieved by employing uniform protocols where possible, such as the isolation of the primary CAFs, and also the sharing of patient tissue samples between the two centres. Finally, I'd just like to send out some acknowledgements. So firstly to the NetRF for funding this programme and for allowing us to present it to you here today, to Professor Irvin Modlin and Wren Laboratories for their work on the NET test, to Martin, Krista and Dal and the rest of the team at UCL and the Royal Free, to Richard and Leo and the rest of the team at Erasmus, and finally to Chrissie and Amy. So thank you for your time, and we'll be happy to answer any questions.
I'm interested in understanding how, if you have any plans of imaging the cancer-associated fibroblasts. You know that there are methods of imaging them with nuclear medicine tracers. Do you have any plans?
No, so far we have not planned for that yet.
But that sounds
but
interesting.
Yeah.
Well, we can talk.
We will talk. We will talk.
Have you phenotyped your CAF? I mean, are you sure that you can reliably call them cancer-associated fibroblasts? Are they activated?
So currently we are looking at using 4 markers for this characterization, and that would be vimentin positive, a filament protein, alpha SMA positive to show that they are activated, and also CD31 negative to rule out endothelial cells, and also E-cadherin negative to rule out epithelial cells as well. So there isn't really any kind of set defined markers for activated cancer-associated fibroblasts, but based on literature, this seems to be what the majority of people are using.
Basic question is there any relationship between mesenteric fibrosis and carcinoid heart disease signaling?
No. So there is, in terms of mesenteric fibrosis, obviously with serotonin levels, there's an association with mesenteric fibrosis. With high serotonin levels, there's an association with carcinoid heart disease. But there's no concordance between having mesenteric fibrosis and carcinoid heart disease.
One more question from online audience. Patricia Dahia asks, she says, thanks for the great presentation. It looks like many susceptible genes end up converging on 2 or 3 main pathways, and that those are particularly relevant to the growth of adrenal medullary chromaffin cells. And so is that an interpretation correct?
So we have seen several of the same kind of pathways being upregulated across the different tissue types. Whether that is specific to certain cell types, we haven't looked into that yet, if that's the kind of premise of the question. But it's interesting to see if we could maybe isolate certain cell types within the tissue and test those for upregulation of genes.
Yeah, hi, I really enjoyed this talk. I wondered, do you have any plans to do single-cell RNA-seq of the CAF populations? Because, you know, in other cancers in the CAF populations, well, I mean, there are subtypes of the CAFs, and they can give you a lot of insight into the progression.
Okay, we haven't decided on doing that yet, but we will definitely be interested in doing that to actually see the heterogeneity as well in the populations. Yeah.
Ramesh and then Eric.
So in many studies, fibrosis and scarring converge on TGF-beta signaling. Have you seen that kind of signal in your GSEA studies? And if so, any particular insight into why SINETs in particular might be inducing that kind of signal?
So we have seen a lot of different pathways that are influenced by TGF-beta, but not specifically TGF-beta in itself. So we are still looking at further evaluating those pathways in vitro as well. Those will be our next steps to try and understand better what's going on between the crosstalk as well.
Yeah, very good. Very nice work. These mesenteric masses are really big. How do you know, or how do you choose what part you're studying? You could be studying the lymph node metastasis, you could be studying the calcified component, you could be studying the softer component. How do you know what you're studying with your transcriptome analysis?
So at the moment, we use pathologists who will dissect the tissue for us and decide parts of the tissue that we will use for our analysis. We have discussed potentially going into things like microdissection or staining and making sure that we have, for example, 80% of the cells or of a certain cell type within that tissue, and that's something we can look forward for in the future.
So next, it's a great pleasure to invite Pedro Marx to come and present from Lisbon, who's going to talk to us about the role of CCL2 and IL-8.
Okay, so good morning, everyone. First of all, I'd like to congratulate the Neuroendocrine Tumor Research Foundation for organizing such a beautiful symposium. It's my first time attending, and also thanking for funding this research on the microenvironment of pituitary and endocrine tumors. I think it's one of the first projects, if not the first, that this foundation is funding on pituitary, and I'm very happy for that, because it will allow us to develop the knowledge on microenvironment on such rare tumors and remains still very large unexplored. So you might not be extremely familiar with the pituitary and endocrine tumors. This is a very important tiny gland that controls the great majority of the hormonal activity and endocrine organs of our body. It's located in a very small space, but it has very important neighborhood structures, such as the optic chiasm sitting above, which is responsible for our vision, or laterally we have also the cranial nerves that are responsible for the moves of our eyes. The pituitary endocrine tumors are actually relatively common. They correspond to about 15% of all intracranial tumors, although the clinically relevant PIT-NETs prevalence is about 1 in 1,000 people. These are benign tumors, but they actually create huge burden to patients due to mass effects. For example, they can compress the optic chiasm, leading to visual impairment or blindness, as well as hypopituitarism, so meaning loss and deficiency of the pituitary hormones and leading to huge hormonal issues of patients, as well as hormone excess in case of functioning tumors that hypersecrete, for example, growth hormone, leading to acromegaly, or ACTH leading to Cushing syndrome, which are conditions that brings huge problems to patients, including morbid mortality from the cardiovascular point of view. There are a few markers that have been studied and validated for predict tumor aggressiveness, such as cavernous sinus invasion and Ki-67, but they still remain poor markers, and we are desperately needing for more markers, either for predict aggressive disease and also for identify targets for treatment. The microenvironment of PIT-NETs remains largely unexplored if you compare to other cancer types. However, there are few recent studies showing that this might be a relevant part of the tumor. Some mesenchymal cells have been studied, as well as non-cellular components such as cytokines, chemokines, and growth factors. And a few years ago, we did this study. We screened the cytokine secretome of primary cultured human PIT-NETs, and we identified as the top 2 secreted molecules are of 42, IL-8 and CCL2, and that's why these are the ones that we are now focused on this project. The PIT-NETs are also widely infiltrated by immune cells, particularly macrophages, which are typically attached to the microenvironment by the action of chemokines such as CCL2 and IL-8. So the aims of our study is to identify the role of CCL2 and IL-8 in defining the clinical phenotype and outcomes of patients with PIT-NETs and their usefulness as potential diagnostic and prognostic markers, as well as therapeutical targets. So we have a cohort of 116 cases with paraffin blocks and SNAP frozen tissues. Because we focus on cases where we have both samples available for RNA and protein studies, we ended up with 86 patients who had pituitary surgery at our center between 2014 and 2020. And then we looked to the expression of CCL2 and IL-8 by RT-qPCR and immunohistochemistry. And then we went on to correlate with clinical, pathological, and outcome data of our cohort of patients. So here's just the baseline clinical features of our PITNET cohort. I'm not going into detail in each one of them, but just to say that we have roughly half cases that were females, 52%. Age at diagnosis was 55, and then we have different clinical features that we also checked. About the clinical outcomes and treatment-related parameters, these are the ones that we have looked. As you can see before the last line, only 3 patients have been deceased, and not really to PITNET causes. So this is not a tumor that really killed the patients, but as you can see in many of these parameter features, they can be very resistant to treatment and difficult to treat, with some cases requiring radiotherapy, multimodal, multiple treatments, and still a high percentage of cases remain with active disease at last follow-up. So we really need to do more for patients who are not cured by a single surgery. So here you can see the expression data of CCL2 across the different PITNET subtypes. So we essentially have 3 subtypes of tumors: acromegaly, which are growth hormone secreting tumors, non-functional pituitary tumors, and Cushing, which are secreting of ACTH. We didn't see differences in terms of mRNA expression of CCL2 across the types, but the Cushing cases had significantly lower CCL2 immunoreactivity comparing to the others. So in our cohort, we identified higher mRNA expression levels of CCL2 in males, in patients who had hypopituitarism at diagnosis, and also interestingly in the cases that required more often post-operative medical therapy, in cases who require multimodal treatment, and in cases who had active disease at last follow-up. So it seems that the cases that initially expressed higher levels of CCL2 may somewhat behave more aggressively and more refractory to the treatment of surgery. We also found a positive correlation between CCL2 expression levels and the total number of treatments, and also an interesting negative correlation between serum cortisol and CCL2, and it's well known that serum cortisol have anti-inflammatory and immunomodulatory roles, so these might be somewhat related. We found positive correlation between CCL2 expression and serum IGF-1 at last follow-up in acromegaly patients. So IGF-1 is like the hormone that we checked to assess the activity of the disease, and there is a positive correlation between these two. In terms of the CCL2 immunoreactivity, consistently we found more expression in males and also in patients who had hypopituitarism at diagnosis. Also more expression levels of CCL2 in patients who had supracellular extension, and also in patients who had hypopituitarism at last follow-up, and less reactivity in patients who required multiple treatments. We found positive correlations between CCL2 immunoreactivity and maximum tumor diagnosis, and also with the number of pituitary deficiencies at last follow-up. So again, more CCL2 predicting or showing that tumors have some more aggressive baseline features and also poor responsiveness to treatment. We went to look on the correlations between CCL2 with the amount of PITNET infiltrating macrophages, and CCL2 is one of the crucial chemokines to attract macrophages, but not the only one. So this may explain why we found no correlation between these two. Interestingly, we found negative correlation between CCL2 and the expression of CDH1, which encodes icadirin, which is a key protein for the epithelial to mesenchymal transition. So CCL2 seems to lead to icadirin downregulation and therefore to an activation of the EMT pathway, which is a pathway important for tumor aggressiveness and evasiveness. So as conclusions, the CCL2 mRNA expression did not differ across the PITNET subtypes, but ACTH-OMAs had lower CCL2 immunoreactivity than non-functional tumors. Interestingly, we found higher CCL2 mRNA expression levels in patients who had hypopit at diagnosis, had lower serum cortisol levels preoperatively, required more often postoperative medical therapy, multimodal therapy, had active disease at last follow-up, increased the number of treatments, and increase in serum IGF-1 at last follow-up in acromegaly, and increased CCL2 immunoreactivity in patients who had hypopituitarism at diagnosis, supracellular extension, larger tumors, hypopit at last follow-up, and also had increased the number of pituitary deficiencies. CCL2 correlated negatively with icadirin expression, so they may play a role in EMT activation. And I didn't show you the data because we found practically no relevant associations with the IL-8 and clinical pathological and outcome data in our cohort. So overall, our data suggests that CCL2, but not IL-8, may have a relevant role in the pit nets, particularly leading to more aggressive, refractory, and difficult to treat disease. And now we'll do some further in vitro studies to further pursue these projects. I'd like to acknowledge my research group and, of course, the funding, the NET Research Foundation, for supporting this research. Thank you very much.
Obrigado.
Really interesting talk. So what do you know about the receptors for CCL2, and are they expressed on these tumor cells? Are we having an autocrine effect, or what's going on? And I don't know anything about the signaling pathways, if you can comment on that.
So the CCL2 has never been studied in pituitary, neither the CCR2 receptor. We had some preliminary data showing that it is indeed expressed. So, of course, it's very important for this crosstalk between the chemokine and its receptor. Although CCL2 can also activate and acts through CCR3, CCR4, CCR5, and CCR5 is also expressed in pituitary. So this is a difficult, is not a binomial relationship. Of course, after proving this usefulness of CCL2, we'll be looking to some of the receptors and also targeting drugs for the type of receptors. So we have like the plans to use anti-CCL2 and possibly also some specific drugs for the receptors. We'll try first with in vitro and then in vivo if we found exciting data from that. But there are also anti-CCL2 drugs already being used in other solid tumors like carlumab. It's a drug that has been tested also against IL-8, but we are not so excited with our IL-8 findings. And there are also well-known drugs for example, anti-CCR5, which is Maraviroc, that is used for HIV. So there are some potential usefulness for this kind of drugs. And of course, we need to understand the chemokines and the receptors. But again, from the literature, we have no clues because it's nothing yet done in this respect. There are some data on gliomas, medulloblastomas showing potential biological role for CCL2 and CCR5 and 2, but nothing in pituitary yet.
Could you tell us what proportion of patients have the more aggressive phenotype and the ones that need the multimodal treatment? What sort of proportion is that?
So that was the one that I skipped quite quickly. We found like it's difficult to measure clinical features suggestive of aggressiveness. We tend to see the cavernous sinus invasion and higher Ki-67, and this in our cohort was about 30 to 40%. Okay. In terms of patients who require multimodal and multiple treatments, depends on the parameter, but is about a third of the cohort. Okay. And 16%, for example, of the cases required radiotherapy. So it's also quite a good number for a tumor that is benign, and you would expect that an operation should cure the patient. Still, 17% in our cohort almost needs radiotherapy, and about 20% required medical treatment afterwards. So we are talking essentially about somatostatin analogs that are also used in pancreatic and gut neuroendocrine tumors, but we also use them in pituitary, and still some of these parts, like roughly a third, behave a bit more aggressively and resistant to treatment indeed.
Just a question about the in vitro models that you might use, because we're having a lot of thought and discussion around models, and do you have reliable models, and could you just give us a little information about that? Then I'll hand over to James.
Okay, so pituitary is very challenging from the point of research because the tumors are very tiny. We cannot have normal tissue to compare because we're not obtaining specimens from people. There are sometimes possibility to get specimens from people that died on traffic road accidents, etc. But still, is normal. It's not normal gland. It's like gland that has been through a pathology. So our in vitro models will be on some cell lines, and also we'll do primary cultured. We will do 2D, and we are also in parallel trying to optimize some 3D cultures, and we're looking. This meeting was good because I was also hearing about organoids, so we probably will test, and then we'll test like recombinant CCL2, anti-CCL2 to see the effects of these chemokines and its blocking, and then if it works, we plan next to put the primary tumors on the mice models, so that will be after validation in normal cell line work.
An interesting thing about these tumors is they're clonal, and so they kind of arise out of single cells in most cases, and yet they can have these arise from different cells, somatotrophs, corticotrophs, these other types. And I did see where you said it didn't matter what they were for the increase in expression of CCL2. But Lauren Fishburne wanted some clarification. Do you know which cells in the pituitary express this? And if it extends to a small set of functional pituitary tumors, corticotrophs are usually the most aggressive. Most of the cohort were NF, which I assume were gonadotroph tumors, which are usually not as aggressive. And so if you got anything further comment on that.
So the issue with pituitary is that the tissues are always very small, and we don't have micro dissection or something that will really tell you that, okay, these are only pituitary tumors. We know that the other components of the tumors are very small, like very few numbers of macrophages and other immune cells. They practically have no fibroblasts in the tumor core. So we believe still the most of the expressions are coming from tumor cells. Also, there's no tumor tissue in those specimens because in immunohistochemistry, our pathology just assesses tumors areas, and usually the surgeon try to dissect the tumor, preserving the normal gland. So we think that most are tumor cells. About their origin and correlation with aggressive, or we don't have carcinomas in our cohort, so it's only benign tumors. So it's difficult to tell if we have like tumors that are biologically more aggressive or not. So we usually define aggressiveness in pituitary tumors. Those are actually relapse after standard treatments, because it's not like other cancer types, or we cannot say like Ki67 of 20% or more, it's a carcinoma. No, carcinoma in the pituitary is like only those that have metastases, and we don't have carcinomas in our cohort. So the guys are all adenomas.
Next, we're going to hear about defining distinct molecular subtypes of high-grade neuroendocrine carcinomas to predict therapeutic vulnerabilities from Allison Stewart and Carl Gay from MD Anderson.
Hi, thank you very much. I'm excited to have this opportunity to talk to you about some of our recent work on high-grade neuroendocrine carcinomas. So in contrast to what we've heard over the past 2 days, our lab studies primarily the aggressive, poorly differentiated high-grade neuroendocrine carcinomas. So the majority of these arise from the lung and have most commonly small cell histology, but there is a rare population that derive from extrapulmonary sites. So because these are so rare, there are not very many evidence-based treatment paradigms, and patients are treated primarily similar to small cell lung cancer patients. So one of the reasons for this is the lack of molecularly profiled patient cohorts and cell line and xenograft models. So our initial goal was to establish a system to obtain extrapulmonary high-grade NETs, both patient liquid and tissue biopsies, as well as to derive new cell line and xenograft models. To do this, we leveraged a platform that we developed initially for small cell lung cancer, and we've now expanded to include extrapulmonary high-grade NETs. So patients are identified, consented onto our established lab protocols to collect liquid and tissue biopsies. We collect the samples, we put these into mice to establish xenograft models and hopefully cell lines, and then we have a team to do molecular analysis as well as a institutional neuroendocrine working group. So our goal is to identify patients that are receiving standard of care or are consented onto clinical trials, which allows us to collect longitudinal samples from these patients. So over the course of the past year, we've collected from over 30 patients from these various extrapulmonary sites. We've established 7 PDX models and 2 cell lines. And I want to point out this thymic neck PDX. So this was actually derived from circulating tumor cells and not from a tissue biopsy. So this suggests that some of these extrapulmonary high-grade necks actually have high numbers of circulating tumor cells, similar to what we see with small cell lung cancer. So once we have collected these tissues, our goal is to test our hypothesis that these high-grade necks, regardless of the primary site of origin, may be classified into molecularly distinct subsets with unique therapeutic vulnerabilities. And to do this, we've modeled it on a system that we used for identifying subtypes in small cell lung cancer. So in small cell, we used an unbiased approach using non-negative matrix factorization to identify that there were 4 unique subtypes. 3 of these were classified based on expression primarily of 3 transcription factors, ASCL1, NeuroD1, and PAU2F3. And the 4th subtype is a little different because it doesn't express the transcription factors, instead expresses immune genes, and it has an inflammatory signature. Therefore, we call this the inflamed subtype. This inflamed subtype in the Empower 133 clinical trial demonstrated improved benefit over the other subtypes with the addition of immunotherapy to chemotherapy. So we then applied the same 1,300 gene signature to publicly available high-grade neck data sets. So at the top, there's pulmonary large cell, esophagus, cervical small cell, and GI necks. And instead of identifying that there were 4 subtypes, we found that there were, in fact, 2. One subtype is characterized by expression of the same transcription factors, ASCL1, NeuroD1, and PAU2F3, but the other subtype, instead of expression of these transcription factors, we found high expression of another transcription factor, YAP1. This YAP1 high subset we've been able to look at more in the pulmonary large cell cell lines and databases, and we found that high YAP1 correlates with sensitivity to MEK inhibitors and CDK4/6 inhibitors. Additionally, this YAP1 status, we've looked at a relationship to neuroendocrineness, EMT, and high YAP1 is consistent with lower neuroendocrine gene expression and also higher EMT score. And this has been looked at in both cell lines and in patient tumor samples. So high EMT means that they're more mesenchymal cells. Additionally, for the samples that we collected at MD Anderson, we performed single-cell transcriptional profiling for a number of the patient biopsies. So this is looking at the cancer cell populations only. So we've removed the immune cells from these plots. And at the top, this UMAP plot is color-coded by the patient or the site of cancer. And you can see that even though these come from lots of different sites, there's similar transcriptional patterns to the cells, which we thought was promising for our work. Across the bottom, the blue cells indicate positive expression of these genes. And you can see that ASCL1 and NeuroD1 expression are in similar cell populations, whereas YAP1 expression is in very different cell populations, particularly compared to ASCL1. Similar to what we saw in the pulmonary large cell data, we see increased EMT score, so that's colored red, so the more mesenchymal cells are also YAP1 positive. So currently, our goal is to classify the subtypes for the PDXs and patient samples. To do this by performing IHC for these transcription factors, bulk RNA-seq, as well as genomic analyses. And we're very interested in determining whether or not YAP1 status actually predicts response to MEK and CD4/6 inhibitors in these high-grade neuroendocrine cell lines and xenograft models that we've established. And our real goal is to look forward at cell populations that emerge upon relapse in patients, and to understand how we can either target these cells or prevent them from emerging in the first place. And some of the ways that we look at this is looking at intratumoral heterogeneity. So this is looking at how different cell populations are within the same tumor. So if cells are distinctly transcriptionally different, this makes it even harder to target these because certain cells within a tumor may respond to a treatment, but then others may not. And when we look at the high-grade NET samples that we have single-cell data on so far, we see that there is quite a variety of intratumoral heterogeneity in these. Additionally, we're looking at drug-tolerant persister cell populations and what pathways are associated with these by GC analysis. So I'd like to thank Carl Gay, who has led this research, as well as everyone in the Byers lab and our bioinformatics team led by Jing. And of course, the NetRF and the Martha O'Donnell Pagel Research Award, which has allowed us to work on extrapulmonary high-grade NETs, and of course, patients and their families.
What were you saying about platinum at the end and YAP1?
What do we know about platinum and YAP1? So with our work in pulmonary large cell, we don't actually see a difference in platinum response in cell lines. So what we know is that in the patient samples that we look at by single-cell RNA-seq, we do see YAP1 populations following relapse. So that's something that we're also looking at.
Really nice talk. We actually study sarcomas, which are more mesenchymal, and we are also studying YAP, and it's correlated with CDK4/6 and MEK inhibitor sensitivity. Yes. So there seem to be a lot of parallels, which would be great to, I guess, further discuss as one thing. And then my other question is, are you trying to consider TEAD inhibitors, which are relevant to inhibiting YAP activity in your studies?
So we've looked primarily at this in the pulmonary large cell because we have more cell lines on hand. And we've tested, I don't know how many YAP1 and TEAD inhibitors, and honestly, they don't work better in the YAP high subset, which is surprising. I mean, no, we've tried combining them. Yeah, we've tried combining with things because we thought maybe if we combined them, but even that didn't really help. So yeah, it was frustrating because we've tested a lot.
Yeah, we work on YAP some as well, and we found a distinction between cytoplasmic YAP and nuclear YAP. And so it seems to follow drug sensitivity. Have you had a chance to look at that?
Yeah, so we've done a lot of IHC. So most of our data on IHC right now is in pulmonary large cell. And in that, we've only been looking at the nuclear localization. So we tried to see whether or not it would predict whether the patients were treatment naive or relapsed, and we didn't see a difference there. Beyond that, I don't think we've looked at that, but I guess we were only trying to look at nuclear YAP in our analyses.
It's intriguing that your analysis of neuroendocrine cells from multiple different tumors show more similarity than you might predict from their organ origins. Do you have some insight into what the unifying theme might be in the neuroendocrine lineage there? They're quite diverse cell types, right? The different organs.
Right. I think, yeah, I agree. Because even when we originally started, we looked at small cell and then we threw in some large cell with it. And I was expecting them to be quite different, and they really weren't. And then when we added these other extrapulmonary sites, so I'm not sure. One other thing I had actually failed to mention is in small cell lung cancer, we don't see YAP1 expression. It's not expressed ever. So that's why these extrapulmonary and the large cell we thought were very different, because we do see these YAP populations. But so I don't know if maybe, it's not that it all comes back to YAP or anything, but I think there probably is more similarities with these extrapulmonary and the large cell pulmonary large cell that we see compared to small cell? I think they're more distinct from one another.
Just one comment, actually, in small cell carcinomas from other organ sites, a lot of times there's TTF1 expression, which also occurs in the lung small cell. There's some that don't express that and some that do, so there probably is some relation. But my question was actually about the YAP1 tumors. Are there any differences in terms of prognosis or expression or proliferation compared to the other?
So so that's a good question. We looked and we didn't see that there was a difference in survival in an initial data set, but we're trying to expand that more into all of the patients that we've collected from so far. So I don't have an answer yet, but one thing we did notice with pulmonary large cell is that the YAP1 high population has a significant smoking exposure compared to the YAP1 low. And that's honestly the opposite of what we had expected to find, because in small cell, the smoking exposure is much higher and they express ASCL1, NeuroD1, and PAL2F3.
It's a real pleasure to introduce our last speaker for this session, who's Xavier Qianjin from University of Chicago. I think he's presenting virtually, isn't he? Yes. So he's going to be talking to us about all-trans retinoic acid radiosensitization in neuroendocrine tumor cells.
Hello, I'm Xavier Qianjin from the University of Chicago. I will talk to you today about ATRA and how it radiosensitizes neuroendocrine tumor cells via PIN1 inhibition. I'd like to thank the Neuroendocrine Tumor Research Foundation for the opportunity to speak today. These are my disclosures. Neuroendocrine tumors are growing in incidence. They have a high rate of metastatic disease. 50 to 60% of patients when they present have metastatic disease. And PRT is one of multiple therapies that is available for treatment of metastatic neuroendocrine tumors that are well differentiated with somatostatin receptor expression, and one of the most promising one as well. However, PRT remains palliative at this point, and there are definitely differences in how tumors respond to PRT. So the question that many researchers are asking themselves is, how can we improve PRT effectiveness in neuroendocrine tumors? The things that we are looking at in the lab are to administer PRT with DNA-damaging agents such as PARP1, as well as discover new pathways that are associated with DNA repair that could potentially be inhibited to radiosensitize these tumors. So as a basic graph, when you have radiation, it induces DNA double-strand breaks. The cell or tumor cell tries to repair this DNA damage either using the homologous recombination or the non-homologous end-joining pathway. Our lab focuses on inhibiting DNA repair in tumor cells. Therefore, DNA damage would accumulate and the tumor cells would die. The homologous recombination pathway is the preferred pathway for DNA double-strand break repair, and BRCA1 and BRCA2, as a reminder, are important proteins involved in this pathway. Last year, we published our paper that was funded through NetRF and the 2020 BTSI NANETS Award. And we essentially showed that MEN1 function impairs DNA repair in pancreatic neuroendocrine tumors, especially the homologous recombination pathway and the BRCA2 transcription and protein levels, and that combining MEN1 knockout cells with radiation and PARP1 inhibition induces synthetic lethality. So we asked ourselves the question, what about all these other neuroendocrine tumors that don't have MEN1 mutations? 50% of PNETs, and then almost all small bowel or lung nets. How can we radiosensitize these tumors? And we looked at the mechanisms of how MEN1 affects homologous recombination gene transcription. And we saw that estrogen receptor alpha seemed to be an important factor that is transcribing these genes together with the MEN1 ERE MLL complex. PIN1, on the other hand, phosphorylates estrogen receptor alpha or independently BRCA1 and can therefore also affect DNA repair, and PIN1 can be inhibited by ATRA, as I will show you. So PIN1 targets the serine threonine motifs to interconvert between cis and trans states. It's involved in many cellular mechanisms, including cell cycle checkpoints and apoptosis. Depending on the substrate it phosphorylates, it can activate or inhibit oncogene or tumor suppressor genes. and it's upregulated in multiple cancers. One paper, it also stabilizes BRCA1 and helps with the homologous recombination pathway, similar with the estrogen receptor alpha, which is phosphorylated by PIN1. And PIN1 maybe even has a role to play in the non-homologous end joining pathway. This previous study, published 2 years ago, looked at breast cancer cells, and here you could see that inhibition of PIN1 increases DNA damage, as seen by gamma H2AX, but this was only valid for BRCA wild type and not for BRCA mutant breast cancer cells. The authors then further showed that combining PIN1 inhibition with olaparib, which is a PARP1 inhibitor, has a synergistic effect, similar to what we've shown with our MEN1 knockout cell line before. So ATRA, or alpha-transretinoic acid, is a vitamin A metabolite. It binds PIN1 and degrades PIN1, and it's also been shown to inhibit breast cancer cell growth, as well as being an effective therapy in lung cancer, breast cancer, and leukemia in preclinical models. We first started our journey by looking at PIN1 expression in 3 different cell lines: the QGP1 pancreatic neuroendocrine tumor cell line, GOT1 small bowel neuroendocrine tumor cell line, and H727 lung neuroendocrine tumor cell lines, and saw that PIN1 messenger RNA level were at least as high or higher than the breast cancer cell line MCF7. Looking at messenger RNA level in tumor samples from PNET and small bowel nets obtained from surgery, we saw good expression. When we did IHC in these samples, we saw that pancreatic neuroendocrine tumor samples in 90% and small bowel neuroendocrine tumor samples in 50% had either intermediate or high PIN1 expression, suggesting that this is a viable target. We then used 2 pancreatic neuroendocrine tumor cell line and a lung neuroendocrine tumor cell line, gave them ATRA, 100 nanomolar, which is a relatively small dose. And even though we could see a direct effect on ATRA on cell numbers, the effect was particularly hefty when combined with IR, as you can see here, suggesting that ATRA indeed radiosensitizes these cell lines. We wanted to see whether this was due to PIN1, so we knocked down PIN1, and here we see again, similarly, that there's a radiosensitizing effect through PIN1 inhibition in all these 3 cell lines. When we combined talazoparib, a PARP1 inhibitor, with ATRA plus IR, we saw that there was a significantly lower viable and total cell number cell count compared to talazoparib plus IR alone, suggesting again that ATRA has a synergistic effect with IR and talazoparib. We then looked at PIN1 messenger RNA expression in ATRA-treated QGP1 cells, and we found, like previously reported, that PIN1 levels don't change, PIN1 messenger RNA levels. ESR1 levels don't change. RAD51 levels didn't change with ATRA plus IR, but they did increase after IR, which is expected because of increased DNA repair. But we did see a significant difference in BRCA1 and BRCA2 messenger RNA levels, showing that there was a 20 to 60% decrease in ATRA plus IR versus IR alone, suggesting that ATRA plays a role and affects BRCA1 and BRCA2 gene transcription. When looking at protein levels, as expected, we saw that ATRA plus IR decreases PIN1 levels, that ATRA also decreases BRCA1 levels, and increased gamma H2AX levels as seen in this protein, which shows that the IR plus ATRA group has more DNA damage at baseline. And the details are not shown here, but we also did immunofluorescence, and we saw a similar trend that gamma H2AX is vastly increased in the IR plus ATRA group. So we then did a QGP1 mouse xenograft model, which we gave ATRA 1.5 mg per kg IP for 5 days a week. And here again, we saw when you compare the ATRA plus IR compared to IR alone group, that at day 31, there was a significant delay in tumor growth in the latter, with a mean tumor volume that was half that of the IR group alone. So ATRA radiosensitizes neuroendocrine tumors, as we have shown in this presentation. PIN1 is a viable target for therapy as it is expressed in these tumors. The enhanced radiosensitization is likely due to PIN1 that affects DNA repair factors like BRCA1 or BRCA2 gene expression or stabilization of BRCA1 protein expression. Combining ATRA with PARP1 inhibitor has a synergistic effect, and these findings suggest that ATRA is affecting the homologous recombination pathway in these neuroendocrine tumor cells and therefore should be further studied. So the estrogen receptor alpha effect on homologous recombination gene is another topic we're studying in the lab. And here's a poster we presented at NANETS a couple of weeks ago if you're interested. And lastly, we're going to further study how to elucidate the PIN1 mechanisms, especially the effect of PIN1 inhibition on estrogen receptor alpha and on HR gene transcription. We will also study the interaction between ATRA and estrogen receptor alpha inhibitors, such as fulvestrant, and we will use an animal model to study how ATRA works in combination with PRRT. We're currently working on this metastatic mouse model and hopefully should have it up and working very soon. I'd like again to thank the Neuroendocrine Tumor Research Foundation for the opportunity to speak today.
So thank you all for a fantastic session. It was quite an eclectic mix, so I don't think we're going to draw some overall conclusions in our discussion, but it's a really, really interesting work that you've all been working on, so thank you for that.
Two questions, I think. The first was you gave numbers on the aggressiveness, which sounded a little bit high. So I guess your denominator is of those that had surgery, just to clarify that.
Yeah, so these are all patients operated for the first time in our center. So all of those that require further surgeries and etc. Are a bit refractory to the theoretically curative surgery. So they are a bit more, we cannot say really aggressive, but more refractory, more difficult to treat, let's say in this way.
But my question was whether or not you looked at some of the genetic drivers that we know, like AIP, MEN, to see how they correlated with your phenotype.
Yeah, we actually had 2 patients screened for AIP because they were fitting the criteria. They were younger onset of cases. All the rest, no, they are, I mean, they might be, I cannot say that are not, but they sound like sporadic tumors. So the mean age of our cohort is 55, so most of the cases were above 30, and none of them had extigma that suggested MAN, like hyperparathyroidism or other types of neuroendocrine tumors. So we think that we have a totally sporadic cohort, but might be 1 or 2 genetic cases that we are not indeed aware of.
We do a lot of genetic testing on pituitary, and growth hormone is not a requirement for having AIP, and a young age is not necessarily, so it may be present.
Yeah, although we know that AIP positive tumors, they tend to be below age 30, and most are somatotroph tumors. Even the non-functioning tumors, they tend to have staining on growth hormone and at the pathology. I worked with Professor Matt Corbonitz in, I think, the largest cohort of AIP tumors is the International Fibrosis Consortium. We published it in JCM, like a cohort of 170 cases, and practically all of them, I mean, we had 2 or 3 pure prolactinomas, but most were indeed GH secreting tumors, but not all.
I have an online question from Matthew Myerson to Qianjin. Matthew asks, can you comment on the general topic, why so many germline mutations occur in pheochromocytoma with such potentially distinct pathways?
We pick TMEM127 and RET as a typical kinase-related tumors to do the single-nucleus RNA-seq. And then I think the reason why they are sometimes, they have some similarities because they maybe share the same cell origin, but they also have the difference. But I think the molecular mechanism still not so clear. But I think there should be some connection between the kinase groups, like between TMEM127 and RET kinase mutants. But specifically, the mechanisms we are still unknown. Yeah.
So I have a question for Allison. I was curious how your YAP signature for your aggressive high-grade NECs compares, if you've done it, compares to the YAP signature that Sharon Gorski and her student Kevin Yang published in Cell Reports last year for pancreatic NETs, non-functional pancreatic NETs, because they're very different tumors biologically, and yet it would be interesting to see what kind of overlap or distinctions you have between the 2 YAP signatures.
Right, yeah, we used a YAP-TAZ signature, because we were trying to make sure that it wasn't, particularly with single cell data, if you depend on a single gene expression, then you may lose things because certain, the quality of sequencing for every cell isn't always the same. And so we had used a YAPTAZ target score, but yeah, I'll have to look back at that because I think it would be very useful to look in other nets because, we were just using kind of a general YAP score, so yeah.
I think they found that it was associated with more immune genes.
Right. Actually, we see the same thing. Yeah, we looked both by CyberSort in the pulmonary large cell RNA-seq data set and found that there was increased immune infiltration in those tumors. And then in our single cell data, we're seeing an expansion of like CD8 positive T cells and actually I think B cells as well. And we weren't sure what to think of that, but yeah.
Sounds interesting. Great, thank you.
Nancy Dew has a question for Maria or Harry. It's more of a technical question. She'd like to know if your fibroblasts grow more spheroid-like than tumor cells in 3D.
And the morphology of our fibroblasts is they're spindle-like, so not spheroid at all. Yeah.
I have a question for Pedro. I probably missed it, but did you also mention that your pit nets had significant neutrophil infiltration? Was that right? And do you think there's any connection there with CCL2 since you saw no connection with macrophage infiltration?
Yeah, actually, we were a bit disappointing on the neutrophil story. I mean, I showed a few, but very little. So from all the immune cells that we study, they are very little ones. 2 to 3 at maximum of HPF. So, and there are tumors that have no neutrophils at all. My initial studies in London from a PhD, I found more neutrophils, but in this one we have many tumors that even we found no neutrophils. So actually we are not really doing much on those kind of data. Macrophages, they correspond to about 2 to 5% of the whole tumor cell population in the tumor. So they are significantly in higher amounts, and we are also starting to look on lymphocytes. They are about 1% of the whole tumor burden, but neutrophils very little, and that's why we are not really correlating CCL2 neither IL-8 with these cells.
Justo Castano asks a question again of Harry and Maria. He'd like to know, are CAFs a suitable target for somatostatin analogs and or PRRT?
We haven't assessed the genetic expression of the CAF cells as of yet. We're more looking into the factors that the tumour cells are more secreting that changes the proliferation or the markers that the fibroblasts are showing. But that is something we could look into and see.
Maybe can I just, I think it would be really interesting that you look to the somatostatin receptor profile in the fibroblasts from the, because even we did some studies in our pituitary tumor fibroblasts, and we treat them with the pasireotide because the pituitary they express STR1, and we found marked decreases in many cytokines, particularly interleukin 6. So maybe it would be really interesting to know in the neuroendocrine tumor field. And we also use somatostatin analogs, so it would be nice to know if these drugs have an effect to the tumor cells, but possibly synergistically also to the non-tumor cells like fibroblasts. It'd be really good to see that.
There are new tracers for cancer-associated fibroblasts, and one of them is the FAPI that can be used, and it's used mainly for diagnosis, but there are therapeutic trials. Therapeutic counterparts have to be tweaked a little bit, but trials are ongoing for several cancers with gastrointestinal malignancies and pancreatic, GI, but also neuroendocrine.
So when we do DOTA scans, we of course see the pituitary is quite bright. And so my question was whether or not when you use PRRT, we're not really aware, or at least I'm not aware, of pituitary directed side effects and damage. Are you aware of this as something that we're overlooking? And then the second question was, is there any use for PRRT as a therapeutic modality with these pituitary tumors?
Yeah, surprisingly, there are very little data on such a major topic about the role of PRRT in the treatment of pit nets. There are a few very little series and case reports with some, about a third may respond to PRRT. So I think there's some plannings to further pursue this. I know that this a clinical trial possibly starting in the Netherlands, in Erasmus, to really further dig about the role of PRRT. About pituitary dysfunction, there are one study, as far as I'm aware, that up to 10 to 20% may develop some degree of hypopituitarism, because these cells, they need to express SSTR2 and 5 mainly, but also 1 and 3, and they can actually be affected by the treatment. And that's why they light up in the scan. Most don't develop hypopituitarism or low function, but they might. So it's good to keep particularly like the thyroid axis and also the cortisol axis is quite important to keep an eye on the neuroendocrine tumor patient that may have symptoms of hypopituitarism after PRRT. Possibly should worth to have a look at the pituitary function at the baseline and then follow up.
Just one comment about pituitary treatments with PRRT. So I have experience of treating pituitary tumors when I was in Milan at IAO. We treated a few patients. So there are a few considerations to be made. Yes, of course, it's a scant experience. One consideration is the expression of somatostatin receptors. Not all of the tumors express high amounts of somatostatin receptors. The other thing is that the majority of pituitary tumors are treated medically and they don't even reach that level. When they do, they are usually far advanced, resistant, received already multiple treatments, including radiotherapy with all the effects on the somatostatin receptor expressions. The other consideration to be made is that the amount of radioactivity is to be tailored, because obviously the amount, like 200 millicuries of lutetium dotatate would be more bioavailable to other organs, and very little would be concentrated in tumors. So unlike classic neuroendocrine tumors like gastroenteropancreatic with multiple metastases, where multiple sites take up the radioactivity and less radioactivity is bioavailable for normal organs, here the whole strategy would have to be thought.
So the question is for the group with single cell data. We have found with our data from single nuclei that immune cells tend to be underrepresented with the natural preparation of frozen cells. So immune cells tend to not survive so well the frozen aspects of the process. So I was wondering if the pituitary and the neck groups had single cell, I'm sorry if I missed that, single cell or single nuclei, and whether we can, if it's single cell, fresh cells, then we can really explore better the immune aspects of these samples or not. And then later I'll have another comment because it's a different, from a different topic.
Yeah, actually, what we've learned in our high-grade NECs is that we have to do single-cell RNA-seq like within 24 hours, like we have to do with fresh samples, which sometimes makes it an expensive gamble on where patients are predicted to have one, disease type, and then we find out after pathology that they had something else, and oops. But yeah, we actually get good representation of the immune cells, and we get tons of immune cells. Like, we're excluding more cells than we're keeping in when we look at the cancer cell populations.
Going back to the question about the pituitary function and the pituitary treatment. So we have had some experience with pituitary nets that we have treated. In my experience, the effects are limited because these patients are at the end of the line, have had previous treatment, radiotherapy, and so on. So I think those cases that have been published are probably also by publication bias, because in my experience it's very limited, these effects. And regarding the function of the pituitary, when we started the phase 2 trial back in 2000, with the first administrations of lutetium dotatate, we did weekly measurements of pituitary function, and there was no decline in pituitary function after PRP. The only thing that we found is that there is a temporary decrease in the testosterone, which is primary, so that's the sticky hair node. But no pituitary dysfunction after PRP.
Just one more thing to follow up on the single-cell RNA-seq question. I also don't think I mentioned that all of our single-cell is on core needle biopsy, so surgical resection is like very rare in high-grade NECs, so I don't know if that impacts things for other people doing single-cell. But
I just wanted to really thank all of our speakers in this session. It was absolutely fantastic, so thank you for that.
Great talk. Really great talk.